very nice to be here and to give this speech today. And while they set up the presentation, I was really thinking about we have a big mixture here, the group, the academics, the startups, and as you mentioned, I'm coaching several startup companies. We talk a lot about the personalized healthcare, and today I thought I'm gonna focus here, yeah, a little bit on a story, and that sometimes it takes stamina, sometimes it takes time, it takes passion, and it's not always that easy. And so I decided to dedicate this talk really to what in Germany the saying is. It's actually der Weg ist das Ziel. Directly translated, it's the journey is the goal, or what you also say, the journey is the reward. Why do I say that? It's because very often, and I have basically to start, I was a member of the Herceptin team responsible for diagnostic when the Herceptin was launched ex-US in Europe and in other countries. And we had to battle it through with this diagnostic piece, brand new thing, you know, in many different countries. And then for several years, there was not a lot. There was the Herceptin story, and everybody was referring to the Herceptin story. And people said, okay, this was this one-time shot, and you make this story based because you have this diagnostic, and this is the story you are doing. And then we said no, because this was the first one. And if you think back, it's 25 years ago when Slayman and the people, you know, really started to think about this concept, and it's still very valid. And this is because it really takes time, and it's a huge effort. It is a change. And this is basically the idea where we said, okay, it is an ongoing journey, and there is no basically end goal over the next years or so. It's just stepwise. And let me just start here from where science and medicine is driving us. And I mean, for many, many years, you know, that the old kind of medicine, it was what you smell, what you can taste and taste. Few things basically you can do. And then the last 100 years, you know, it's really started diagnosis, treatment, biochemistry, cellular processes, and then over the last years it really got crazy. And this is just one example which I'm always referring to. Many of you might remember this huge project, just this goal for the whole science to have a human chromosome, a genome basically sequenced and get all the information. And it is only about 10 years ago, you know, when this project really came to an end with 3 billion Swiss francs in total, 2.5 year total time. And, you know, many of the leading institutes were involved into that, you know, doing a tiny piece of one chromosome and then add all the information together. What a huge endeavor, you know, and then you made it. Then a few years later, the James Watson Genome project, you know, then it was still very expensive, a lot of sequencer, but then it was like 60 days. I said, what a difference, you know, what you see. And then a few years later, you know, it's only a matter of days and you're in the range of 10,000. And now, you know, you get this kind of erosion where you say it's 1,000, it's less, it's only a few days. But then again, what do you do with this information? That's the second question. And this is the kind of problem I think we are all facing, because over the last 20, 30 years, you know, this increase of knowledge, be it on the IT side, on the computer side, on the sequencing, on the genomic, but also on the proteomics, metabolomics, you know, that gives us an hint and ideas of pathways, basically how diseases, you know, come into play, you know, what you can do. But you also learn about that, like, viruses are smart, that tumors some kind are smart, outtrick, you know, if you have single treatments, the area of screening, of large screening banks, where people say, now we understand exactly the mechanism, now we can hit very hard, you know, and this is how we stop it. But then you later learn basically that the other system, your enemy is also not dumb, and then you just circumvent, you open a second pathway, and your drug simply does not work anymore. So all of a sudden, you more talk about resistance. You have initially results, you know, and you think, that's it. And then you see the resistance. And then you learn more about the pathways. And then you go into disease mechanisms. And then all of a sudden, the number of plausible targets gets in the range of thousands. And all of you know, if you set your bets in a pharmaceutical company, be it a small company, but even if it's a large company, and if you go into large trials, you only have a limited number of options. And if those fails, you run out of money. So what you're going to do here gets also more complicated. It seems to be easier because you know more, but the more you learn, the more you understand also how complex it is. And This is one of the questions when this came up in this cartoon, the challenge of personalized healthcare. I mean, do we really, get there? What is going to be the future? Where do we use it? And that's for me, it's really right now, it's a matter of perspective. It's really where do we stand here? You can always argue for all of the drugs, it's not the end game. We cannot cure the diseases. But in many of the diseases, these new tools and the development of drugs in combination of drugs really make a difference. And again, it's a journey. We are on this journey, and it's going to be in many diseases a stepwise process. And I just want to bring up this one because this personally really affected me. I was working on my PhD in an industrial lab on reverse transcriptase inhibitors of HIV. And we had fantastic drugs, and at these days, the first science signals came out about ACT resistance and also about resistance to the non-nucleoside inhibitors. And we had a few fantastic compounds, and we stopped it those days because we learned that this resistance is coming so easy. I left that field, I went more into gene therapy and oncology, and years later, when we looked basically how this whole thing develops and how the combination of the different drugs for HIV/AIDS combined with the diagnostics really came into the players that we left too early. We didn't believe into the whole concept, and we got shocked from the primary results. I remember that exactly. We had this pocket of the enzyme, and the non-nucleoside goes in, and then basically you have a change in one amino acid, and then it falls out, and the whole thing is over. And that was these days basically the shocking finding, and we stopped this. And over the years, I am convinced that some of our inhibitors really might, play a real important part here, but we stopped too early because we did not see the whole picture there. And I think it's the same here. This one-size-fits-all approach, I personally and we as a company believe that in many, many diseases it will not work. I also have to be very clear. We are talking in many cases not really about individualized. It is more that you enrich patient groups for better response to certain drugs at this point. It might be different if you really talk about genetic diseases where you really, really know that you have a genetic effect, so then it's individualized. But here for most of the drug developments, we are talking about a strong enrichment to move the drugs over the hurdle and to the next step. And I think in the end it will benefit, and it should benefit in principle, the patients because they get the best treatment at the right time. You have for the physicians and the providers a very good benefit here, and hopefully can also reduce unwanted toxicity. This piece, the regulators, the policy makers, this becomes more and more important. And you might know that, especially for FDA, for most of the drugs, in the meantime, they really ask, do you have a biomarker? Do you have a diagnostic test? It's not the other way around. You have to argue if you don't have one, instead of just going for the all-comers. And payers and reimbursers, of course, all these discussions go on in parallel. And what is the evidence and what are the questions? And this is for me, this is I think a very, very important message here, because sometimes on the political level and in discussions, two very distinct things gets mixed up. The one is access to generic drugs and make drugs cheaper. They run off of patents, make drugs cheaper, make them available to a very reasonable price. And if you look at this slide, what this one shows is these are the number of patients in percentage which initially respond reasonable well to a certain treatment. And if you have, like I say, cardiovascular, blood pressure or some of these diseases where you in principle have drugs where initially 60 to 80% of the patients react to a treatment, I don't think that we need to do personalized healthcare enrichment and this more expensive drug developments. And here, very nicely, the concept of generic drugs which are cheap and which are accessible is very meaningful. Now, but if you go in areas like CNS, like oncology, many viruses, where in principle no treatment is available today, and this is what I always want to tell, then if you just embark on generic drug development which make it cheaper does not change a thing, because in these areas you do not have effective treatments. And here you completely need to think different. Here you need to invest. And like CNS, and you know it from many companies, and also oncology, it's a huge risk if you do all of those trials, because the benefit and the competition is high. And here the personalized healthcare piece is really important. And the big question which we need to sort out basically as a community, as researchers, as academics, where is basically the threshold where you say, you know, the drug is still good, initial drug for all comers, or where do you really need then at the second step this personalized healthcare approach? And that brings me to the second point, and this is what you say, okay, what can we learn from the existing drugs and what does it really need in the future? And if you don't have efficient treatment, if you don't have good responder, you know, it's gonna be it's a moot point that you say, okay, you want to use the samples, recycle them, learn from them, but as soon as you have good clinical efficacy, you know, then it becomes a different story where you can say, okay, what can we learn really actually from the hospitals, from the doctors, from the clinicians, which they are treating patients, they're using our drugs and they see big differences. Sometimes you see very good responses and sometimes you see very fast onset of resistance or non-response. So what can you learn and can you transport this back into the R&D situation where you can really learn and launch second generation or combination of that, which is very often necessary. And I just want to focus on a few oncology drugs right now. I have cycled them from our portfolio and just talk a little bit about what happened over the years. And again, the story starts with Herceptin, and this is a story of about 25 years. 1.4 million patients are treated in the meantime. We have about 20%, 50 to 20% of them are really HER2 positive. So it's a small portion. But initially, the HER2 was really detected as a marker, which was really on the disease onset, and it was a predictive marker that the disease is very bad and there was no good treatment. So that was the starting point, and over the years, it turned around. This is how Herceptin works. So on the mode of action, we've really learned a lot. It also worked somehow in stomach cancer. We got disappointed in other areas where the HER2 receptor is there, but we could not see the same effects. But that was one of the biggest trials ever done when we realized that from the metastatic setting, it might be also beneficial to move it in the early onset of breast cancer. We did a large study, and this study really included, I guess, the screening of 10,000 patients. And all of that was done with a very good diagnostics. We had central labs because we wanted really to make sure on the diagnostic side, if we include the patients, that our diagnostic is really well done. I come back to that point later. And so this is the number where I think we did save over all the years with Herceptin. People would say it is a good start. It is clearly not enough, but if you look at the numbers, it's a pretty big number basically, where you can do the math and calculate on patients which got saved basically with this Herceptin treatment. But then, if you look into that early breast cancer and you just added the facts here. 10 to 15% diagnosed with early breast cancer will go on, and then you have this setting, the metastatic breast cancer. And for this, it's still a very complicated situation, and we need more therapies, we need new therapies. And I just also want to quickly say that the diagnostics here, it was really for many years underestimated. We had some findings. This was from the American Pathology in the middle of 2005 to 7, where still 1 out of 5 patients got either false positive or false negative diagnosed. And they looked deeper into that, and what they realized is that especially small labs, which have only a few patients, you know, that a lot of patients got wrong diagnosed, while the larger labs, which do a lot of training, and in the middle of it, most of them is really accurate. So that was also one of the learnings, because if you look at the distribution of the costs between what you need, what you spend for testing, and the result then, if you treat the wrong patients on the other hand. So this is quite striking, and we have to learn that, and we have to educate and come back to this point. So it's the different testing, we have them, it's the protein, sometimes it's the genetics, it's the fish marker, and in principle now we have a very good diagnostic system. Then we moved on and we brought a second antibody in, and the mechanism is just shown here. So the Perjeta, it's known that they form these dimers, and these dimers are active between the HER family. And while the Herceptin really binds to the HER2, we know that the second antibody, the Perjeta, basically moves between them and stops the dimerization and stops the internal signaling. And that was also a finding over many years, and this was then the result of that, that the combination with Perjeta and Herceptin in the metastatic setting, you know, added another 6 months, which is, again, this is another lift up. It is not the whole story. This is a second step. And with that, you know, we continued and we brought something else in. And that was the idea that, you know, that for Herceptin, you still need the chemotherapy part. And this is basically the toxic part. It's not the antibody. So for many years, the scientists in Genentech South San Francisco tried to model around where you link the toxic part to the antibody so that once the antibody really binds to the tumor site, then you unload and you only create the local effect. And I can tell you they did rounds and rounds and it got too toxic, so it failed. They did it again and did it again, and they did not give up until they came up with the DM1. And this one, that really seemed to work, and then they moved it forward. Again, this is a story of 15 years or more in research and try and failure and retry and move it forward. And then we looked into that, you know, that's the next one, where we said, okay, this one, it's from the convenience, it's less toxic. And again, you raise the bar up, and these are failures for the Herceptin. And then you can add another treatment here, basically, where you again delay here with a reduced risk of death of 32% and improve progression-free survival for another almost 10 months. So what I try to explain is from a disease point where in the beginning there was absolutely no hope or only very little hope, you know, stepwise with the development of the drugs and the learning and the next generation, you go stepwise into a management of a disease over many years on a very good quality of life. That's basically the story we're having here. And this shows you also how this continues. All of that is ongoing, basically the large clinical studies. They go on for the next 2 to 3 years in the adjuvant setting now, and hopefully that again gives us another lift if you then have the combination of Herceptin and you save even more people. Another example here is lung cancer. And when we launched the drug Tarceva, there was some efficacy in lung cancer, but it was clear that some of the patients really reacted well, but we did not know why. And we had the idea because it was EGFR inhibitor, it's a small molecule, it was EGFR, so it had to do something with the receptor. But in the beginning, we could not find it. We did on the protein immunohistochemistry, we did again on the genomic level, but it took us relatively long to find out that it is really the EGFR mutation, which is the most important one. And for those, they are particularly sensitive to treatment with Tarceva. And then what you see, and this is shown here, and this is for end-stage lung cancer, this is also very striking, you know, that it's 31 months compared to about 12 months with chemotherapy in those patients which really harbor this mutation. And now the second part comes here, which was also very surprising, that also in different parts of the world, this mutation in the cancer shows up on a different level. While in the European, so the Caucasian population, it's only about 10% of the patients. You know, in Asia, you see 30% of the patients harboring this mutation. And that's so again, if you look at the complex situation, you launch a drug, and then you think you do it for lung cancer, and then you comes a biomarker, and then the biomarker, even in different parts of the world, is being expressed in different levels. So these are the things, the learnings we have, and in those patients, in the meantime, I think Tarceva is well accepted, and you might also know that this area of lung cancer, you know, there's a lot of work ongoing right now, and there is from like 10 years ago where it was just nothing, you have about 6, 8, 10 experimental treatments where you can really make a difference. And this shows basically that from the origin where you say, okay, this is lung cancer, you move now more towards a biomarker signal pattern where you say if this genetic activation is there, you give this drug, or if this pathway is on, we can go for that drug. And this is really changing over the years, but it's also complex if you think about on the regulatory side how you register these drugs. So for oncology, I think PHC is making its way. It's becoming more and more reality. This is what we are doing. I also included here a viral test here. Herceptin, subcutaneous, Perjeta, Kadcycla. So we are moving these drugs forward in this indication breast cancer, also in lung. And that brings me also internally then to how we really apply that. We have the diagnostics in-house, and it's really about the medical value here of the quality of the test. You develop the biomarker tools, technologies, validated assays. Then at one point in clinical trial, it becomes really very important that you think far ahead, because if your concept really makes it, if you are successful, you not only have to bring the drug to the patient, you also have to provide a test which is worldwide rolled out in all the countries where you launch the drug, and also where you can have the same quality of the testing, be it somewhere where it's very hot or somewhere where it's very cold. So you have to think about it, and the regulatory expertise is important, and also that the commercial reach, that you really get it out. And on the pharma side, again, you can work on your content. On the R&D side, patient sample, clinical trials, and so forth. Now, over the years, what you see here is that we really live this concept within the company. What we do over the years, we really bring the scientists together. We focus on the areas of interest, like virology, like oncology, and they start doing joint projects. And most of them start relatively early in research, where we try to learn about the mode of action or the mode. Why is it a resistance to the virus? What is coming? And this slide basically shows, so research, 250 internal collaborations. Then in development, and I think this is really the striking number, 8 out of 10 of our drugs which move towards the market contain or have the companion diagnostic concept. So 10, 15 years after Herceptin, where we said, okay, this is the one, and okay, got it, fine. This is moving along, but this is the time it takes if you move the concept back into research, do your homework, and then move from research again to the different stages through clinical development. This is the time it takes until you get there. And on market, again, we have now in the meantime, we have 5 out of 25, so it's roughly 20% or so of the drugs are really companion diagnostic drugs. And I would imagine or estimate that it might double, that it might go up to 50%. Probably not all of them, and you don't need to have all of them there, but it's going to be a larger portion of that. But that's not the end. This is the beginning. And what we see right now is, and we dig deeper and deeper into the biology in this area. So the dysregulated cell signaling, this whole area, we try to understand more and more. And this is also, if you think about our melanoma drug, the Zelboraf with the BRAF inhibitors, we realize very good response, but we also see that there is resistance. So the tumor is coming up again. And now we look into ways basically to double block or triple block the tumor. We are getting back a little bit again in this kind of AIDS scenario where you think, okay, what can you do? And then the second question is, what is better? Do you treat in parallel with 2 or 3 or shut it down? Or you first give the first drug, and then if you see resistance, you give the second drug. All this needs to be sorted out in the clinical setting. The micro-tumor environment, there's a lot ongoing in the tumor. Some different cells play a different role here. It was absolutely underestimated. So this is also coming, and maybe from the simple biopsies, where in the past we just looked at the HER2 overexpression, maybe it's more important to see what kind of cells are in there, and if the good guys in or the bad guys in, or how do they communicate, what are they doing there. So that again, this is a new challenge also on the diagnostic side, on the imaging side, on how you basically interpret this data, and also then on the clinical setting, what kind of experts you need basically to make a real interpretation diagnosis out of those data. And then again, the cancer immunotherapy, and I'm a big believer. I think if we can really get away from killing the cells from the chemo, that you just simply follow the concept that the fast dividing, fast growing cells die faster. If you can activate the immune system in this direction, you turn the whole thing really completely around. And these are the areas where we work, where we try to move the next generation. And all these things include diagnostic testing, finding, mode of action is moving forward. The open questions, and I'm come closely to the end here. Quickly now, how will PHC grow? What are the key drivers? What is the key value? I mean, very valid questions, and I probably, with the results of the clinical trials with the drugs, we're going to find answers, and some of them will be very successful, others will fail, but I think it's a part of the game. What areas come beyond oncology? I think for oncology we can prove it. Is this really moving along? If you look at all the neuro diseases, CNS, big problems, it's a different story. So large genetic investigations here, we will see what comes out of it. Virology is clearly there. I think inflammation will come, some others. Multi-gene approaches, sequencing, whole genome sequencing versus single markers or small set of markers. How will this move forward? What are the regulatory paths for that? Then, like in oncology, heterogeneity. doesn't mean that a tumor is a tumor. You know, there are different parts in the tumor, and we know it in the meantime that some of them might have mutated tumor cells, in other areas they are not. How can you be sure that what you diagnose and what you sign as a pathologist is really the status of the tumor of the patient? Big questions unsolved. And again, how will regulatory and reimbursement environment evolve over the years? How can we, you know, work that? So, what I try to tell today is personalized healthcare is here to stay. And I guess there is more to come. The question is when, how, who will do it, how successful, what is the timing? Many open questions. The other diseases, you know, are in evaluation. Asthma, I said, inflammation, CNS. There are still challenges to overcome, and many companies see that. Is also when you start with a small company, if you look into this concept, please make sure that on the diagnostic side you have a robust testing, and that would later what you offer to the company is reproducible, or you still have the samples and can move it forward. Because otherwise you start on a research assay and there's no next step then, because there's no platform. And then you get really stuck if you go into this concept. You really have to think far ahead. If you are successful, what would be the next step on the diagnostics? You know, it doesn't need to be Roche, it can be any company, but you need to have a commercial reach or people who can develop it. Otherwise, you know, you get your surprises that you might have some interesting results on the pharma side, but you cannot really document it on the diagnostic side. So be careful about that one. And for us, of course, to have it in-house, I think it is a strength, and we really try to move this concept forward. This is where we believe the future will be, at least for the drugs, where we're developing in diseases like oncology and in this critical phase, and there's no real alternative to that. And I think as long as we keep that in mind, that we move it forward like we're doing in the breast cancer, it should be a good way. And with that, I want to close and take questions, of course. Thank you.